Dear students, today we shall be talking on respiration in mollusca. We shall be covering it in general and mostly emphasizing on Ctenidia, and then in the next lecture we shall be dealing on the terrestrial mode of respiration. Before starting, it is my request to you all to like and subscribe my channel BioLearnia. First of all, we shall be knowing that what respiration is and what is the definition of the respiration. We simply say respiration is the process of the gas exchange between the air and the organism cells. Three types of respiration that include the external respiration, internal respiration, and the cellular respiration. When we say external respiration, it is the breathing process. Simply, it is the breathing process which involves inhalation and the exhalation of the gases. That is, whatever we inhale and whatever we exhale. Second is the internal respiration that involves the gas exchange between the blood and the body cells. Now, when we talk about the third form, that is the cellular respiration, it involves the conversion of the food to energy. Aerobic respiration is the cellular respiration that requires oxygen, whereas the anaerobic respiration does not require oxygen. So we have the three modes of respiration, that is the external respiration, which is just breathing in and breathing out. The second is internal respiration, that is whatever we have taken in, whatever air we have taken in, it exchanges between the blood and the body cells. The third is cellular respiration, which takes place inside the cell, and wherein the food is converted into energy. Now herein we have the two modes of respiration, that is aerobic, which involves the requires the oxygen, whereas anaerobic does not require oxygen. Now when we talk about the respiration in the mollusca, we basically define it as the process in which the organisms exchange gases between their body cells and the environment. Now, rightly from the prokaryotic bacteria and archaeans to the eukaryotic protists, fungi, plants, and animals, all living organisms undergo respiration. That is, all have to breathe, all have to respire. Basically, all mollusks breathe by gills that are called Ctenidia. Now, these Ctenidia are also called as the comb gills because they look like the shape of the comb. It was the capability to breathe oxygen from air that finally enabled today's terrestrial mollusks, that is the ancestors, to move on to the dry earth. Now, rightly, it was the Ctenidia which was the primary, you can say, respiring organ or the primary organ of respiration. But later on, with the evolution, these very terrestrial organisms, they evolved and they had the capability to breathe on land as well. That is, they took up the oxygen from the atmosphere. This transition happened several times in the separated cases and in different systematic groups of the gastropods. All, however, or whatever their systematic origin, they have in common, they breathe dry air. And mostly we do have the gastropods which breathe on land. We call them as the terrestrial mollusks. This respiration organ is in terrestrial mollusks, this very respiration organ, that is the Ctenidia, that is reduced, but still respiration takes place in the pallial cavity. That is why it is also called as the snail's respiratory cavity. Now it is just a cavity inside the mantle of the, you can say, the snail or the gastropods, which helps in the taking up of the air in and out. Now, when we say the modes of respiration in mollusca, there are three modes of respiration, that is cutaneous mode, Ctenidial mode, and the pulmonary mode. So we do have the three modes of respiration. As the we call it as the cutaneous respiration, we call it as the Ctenidial respiration or the branchial, that is by the gills. So these are also called as the respiration by the gills, and the pulmonary, which act as the lungs in the mollusca. So the cutaneous or the mantle, we also call it as the respiration through the skin. Ctenidia, that is branchial, or we call it as the gills. Pulmonary, we call it as the respiration by the lungs. Now we shall be talking about the types of respiration in the mollusca based on the medium in which they are respiring, the based on the habitat they are taking up the oxygen. Now we do have the aquatic mode of respiration and the terrestrial mode of respiration. In this very lecture today we shall be mainly focusing on the aquatic respiration, which takes place in water, and this oxygen dissolved in water is used, and the surface of the respiratory structure is greatly increased to allow the rapid absorption of the oxygen. So it means that whatever Ctenidia are there, whatever the comb gills are there, they increase the surface area. The surface area is increased so that maximum water comes in contact with that very, you can see, organ for the better diffusion of the gases. The aquatic respiratory structures can further be divided into the primary structure, which is called as the Ctenidia. So we do have the primary structure, that is the Ctenidia. And then we do have the other secondary structures as well, the secondary organs as well, which are used in the aquatic mode of respiration. So some of the organisms, some of the mollusks, they have the anal gills. Some of the organisms, they have the serrata or the dorsal appendages, or we also call them as the pleural gills. So some have the pallial gills, and some have the integument as the respiratory organ, as a secondary respiratory organ. So when we talk about the terrestrial respiration, so when we talk about the terrestrial respiration, we mostly it is also called as the amphibious adaptation, and the adaptation to the terrestrial life that have introduced remarkable modification in the respiratory structure of the mollusk. So earlier they were only in the water, they were having the only aquatic mode of respiration, but later on, due to evolution, they also switched on to the terrestrial mode of respiration, which is helped by certain organs such as nostril lobe. We do have the pulmonary sac, we do have the lungs. So these very pulmonary sacs or the lungs, and then the nostril lobe, that acts as the organs of the respiration on the land. Now we shall be talking about the main organs of the respiration in the case of mollusca. And the first of them, that is the cutaneous respiration or the mantle respiration, it takes place in certain mollusks, wherein these very mollusks, they respire with the help of the skin or the mantle. The skin appears to be the simplest type of respiratory structure that is encountered in mollusks. It acts as a respiratory organ in certain forms where there is no special respiratory device. So in those very molluscans where there is no special respiratory device, this very cutaneous respiration or the mantle respiration takes place. So such type of respiratory organs, they are found in the case of Scaphopoda. We call them as the tusk shells, some aplacophorans, some Opisthobranchia, wherein this very mantle and the skin or the skin respiration takes place. Then in some Vaginulidae, these are the terrestrial forms of the mollusk. In them also the cutaneous respiration takes place. Then in some parasitic forms such as the Antoconcha, the cutaneous or the mantle respiration takes place. Mantle respiration occurs through the whole body surface or the soft skin parts of the body. So it must be very restricted when the body is covered by the shell or the skin is thick and bulky. So the cutaneous respiration, it takes place through the skin of those very mollusks having other respiratory mechanisms such as pulmonate gastropods. So in all bivalves, the inner mantle surface also contributes to the oxygen, although the respiration is mainly branchial through the gills. through the gills. Now If we talk about the cutaneous respiration, in most of the members of the Aeolidae, the dorsal surface of the body is covered with papillae. So in some the Aeolidae, the dorsal surface of the body is covered with the papillae, and the papillae are variable in size and communicate with the heart by veins. So in some cases the papillae which are of variable size, they communicate with the heart by the veins. And in most of the nudibranchia, nudibranchia, they respire through the skin. And in some forms such as neomania, Chetoderma, Aplysia, we do have the dentalium, etc., wherein the mantle is used for respiration. Many times what happens that they directly surpass, they directly ignore the heart, and they directly ignore the gills or the pulmonary sac, and they directly take the blood to the heart. And the cutaneous uptake depends upon the permeability of the skin to oxygen and upon the lower gas tension in the blood and the tissues than that of the external medium. So it is obvious that if the blood is passing under the skin, it is the skin which is very delicate, wherein the blood is coming in, it should be having the lower oxygen concentration than that of the atmosphere, than that of the environment. Now what happens in this very case? When the water comes in contact, or the air comes in contact with the skin, the gaseous exchange takes place by the diffusion, wherein under the mantle, in the blood vessels, the oxygen concentration is less, where in the environment the oxygen concentration is high. So it comes from the environment toward the mantle, the oxygen concentration. Then we talk about the carbon dioxide. It leaves in a similar way, wherein the carbon dioxide concentration is higher under the skin, inside the blood vessels, and in the environment it is low. That is why it moves from the blood vessels to the outside, that is the environment, and thus the exchange takes place, and the harmful gases, they leave the body. Cutaneous respiration is obligatory for those very molluscans which do not have a differentiated respiratory organ. So all those very mollusks which do not have a differentiated respiratory organ, as in this case of Scaphopoda, Aplacophora, and some gastropoda. Now, or it is extremely reduced, wherein these very organs, they are extremely reduced. So in many pulmonate gastropods, although the oxygen uptake usually takes place through the lung, there is reason to believe that a considerable portion of the oxygen is absorbed through the skin. So in genera such as Limnia and Helicosoma, where the skin is habitually kept moist by the secretion of the mucus, animals can remain submerged for a longer period of the time, and particularly at the lower temperature, and during which these very lungs remain closed or they filled with water. When these very lungs remain filled with water or they remain closed, they have no other choice to breathe with the help of the skin or the mantle. So this very respiration takes place in these very organisms which go inside the water to for some time or some reasons. So cutaneous respiration can be studied after inactivation of the respiratory organs as well. So the experiment was done in the case of the some pulmonate gastropods, where in their lungs there was the occlusion of the lungs. So their lungs were filled with the paraffin, so that it can be studied that what is the organ besides the lungs from where the molluscans, these very pulmonate gastropods breathe. So under these conditions, this very residual respiration, it is almost 20 to 50%. The loss of the carbon dioxide occurs through the skin. So in addition, certain amount of the hemolymph after passing through the mantle goes to the heart without passed through the respiratory organ. So it was the Zager and Volvicamp in 1958 who experimented upon the Limnia stagnalis and Planorbis, wherein what they did, they put these very organisms in a beaker which was having the fresh water with high concentration of oxygen. When these very organisms, that is Limnia stagnalis and Planorbis corneus, they were placed inside, after some time what they did, that from a hole from where they took out the water sample, it was noticed that the oxygen in the water sample was consumed, though their lungs, they were filled with the paraffin, that is there were no other organs from where they can breathe. So this led to the inference that there is some organ beside the lungs from where they can breathe. So they came to the conclude that yes, the mental health in the respiration are the exchange of the gases. So in this experiment, the snails were subjected to a gradual decline in the oxygen tension, not unlike what occurs in the natural waters. And Limnia, which has no respiratory pigment in its blood, lived as well as did the Planorbis, which has hemoglobin in the blood. So both these very specimens, they survive. Both these very organisms survived because of the fact that they respired through the gills. So the cuteness or the mental respiration is also prevalent in those very mollusks wherein they don't have the well-defined respiratory organs. Besides, some have the pulmonary mode of the respiration. Now, our main focus today, that is on the Ctenidial or the branchial respiration. So this is the second mode of respiration in the case of molluscans. We call it as the Ctenidial or the branchial mode. The branchia, it refers to the gills, and here in Ctenidia we use the term Ctenidia for the comb gills. Most of the mollusks are the aquatic and respire by means of the gills, or we call it as the Ctenidia. So the aquatic mollusks respire through the Ctenidia, and these are the comb-like outgrowths of the mantle and are located within the mantle cavity. Now herein what you are seeing, that this is the diagram. This is the, you can say, general diagram of the molluscan, wherein you are seeing that this very part which is just comb-like, which is comb-like. Herein what you are seeing, that these are the comb-like organs. This acts as a gill. In the next diagram we shall be able to see that herein water enters, this is the inhalant, and then leaves this very cavity wherein the gills are present. So they come in, the water comes in contact with the gills and then leaves. That is called the inhalant and the exhalant, you can say, system. So in this, the oxygen dissolved in water is used. The surface of the respiratory structure is greatly increased to allow the rapid absorption of the oxygen. Now, if we talk about the Ctenidia, these are paired, symmetrical, ciliated, and two rows of the flattened gill filaments that are arranged one on either side of the long flattened axis, traversed by the afferent and the efferent vessels through which the hemolymph flows. So the Ctenidia, they are paired, they are symmetrical, they are ciliated, and the two rows of the flattened gill filaments, which are arranged on one on the either side of a long flattened axis and traversed by the afferent and the efferent vessels through which this very hemolymph flows. Now, if we talk about the structure of the Ctenidia, Ctenidia are the projections of the body surface or the mantle. This is the Ctenidia. This is here, that it is the representation of the Ctenidia. So they are fundamentally same structure as in the primitive members, such as Polyplacophora, Rhipidoglossa. Then we do have the Protobranchiata or the Cephalopoda. They are fundamentally same structures in all the primitive members. We call Polyplacophora or Amphineura, Rhipidoglossa or Gastropoda, the Protobranchiata or Lamellibranchia, and then we do have the cephalopodons. Then if we talk about the structure of the Ctenidia, this is very interesting one. If we talk about the structure of the Ctenidia, that this very Ctenidium, this consists of the horizontal stem or the axis. So this is called the this is the horizontal stem or the axis. This one, this is the horizontal stem or the axis that is attached to the body, and it bears on either side the row of the arranged delicate flexible, this is the row of the delicately or alternately arranged delicate flexible respiratory lamellae. So these are the lamellae. Now what you are seeing here, that this is the representation of the lamellae here. This is one lamella, this is another lamella, this is another lamella, and this is another lamella. So in plural we call it as the lamellae. So they are delicate flexible respiratory lamellae, or we also call them as the filaments. Surface epithelium of the gill is covered by the cilia. So many times they are covered by the cilia. We shall be just in the next slide we shall be seeing that how they are covered by the cilia. Here in the cilia are not visible, but here in the I have tried to show you the filaments. So the movement of which are responsible uh, for the continuous renewal of the water over the Ctenidium. So this very Ctenidium, which receives the venous blood from the artery through the afferent branchial vein. So we do have the afferent blood vessel, and this is the afferent blood vessel, which helps to take along the deoxygenated blood. That is, this is having the deoxygenated blood, and from the afferent blood vessels the deoxygenated blood is passed on to the Ctenidia. So and after oxygenation in the Ctenidia, so after oxygenation in the Ctenidia, that receives the venous blood from the artery through the afferent branchial vein, and after oxygenation in the filaments it is sent back to the heart through the efferent branchial vein. So what we infer from here, that from here the deoxygenated blood comes, it goes into the Ctenidia, and then it leaves through the efferent blood vessel after oxygenation. Now again, what we are seeing here in this very part, what we are seeing here, that that the blood comes in from the efferent blood vessel and leaves by the efferent blood vessel after oxygenation. So this efferent blood vessel, it is with oxygen, and this comes without oxygen, and the oxygen is renewed here in the Ctenidia. Now what you are seeing here, that you are seeing the passage of the blood in this very direction, and you are seeing the water in the opposite direction. You are seeing the water in the opposite direction. So there, this is called as the counter current mechanism, and you are seeing that the blood is flowing in one direction and the water is flowing in an opposite direction. So this very mechanism is called counter current mechanism, wherein the maximum water comes in contact with the blood so that the exchange of the gases can take place. And another what we see here, that the water flows over the gills in the direction opposite to that of the blood flow in the filaments. So this countercurrent flowing or the countercurrent mechanism, it ensures the more efficient respiratory exchange of the gases. Oxygen of the water passes through the wall of the gill. So this very oxygen, the water which contains the higher concentration of oxygen, that passes the wall of the gill filaments into the blood, and in return the carbon dioxide passes the water. So this is one thing, and the amount of the gaseous exchange is proportional to the respiratory surface area and to the concentration gradient. So whatever the exchange is taking place, that is proportional to the respiratory surface area and to the concentration gradient. So the gas exchange in the mollusk takes place through the simple diffusion, that is without the active transport. So there is no active transport taking place in the, you can say, Ctenidia. Now again, what you are seeing here, that I have tried to show you here some of the creases. These are the creases what I want to show you here from with the help of this the lighter highlighter, that these are the crease on the Ctenidia. So this very crease on the Ctenidia, it helps to increase the surface area of the gills. And secondly, what you are seeing here, that Ctenidia are broader at the on the axial side, that is on the axis, and they become narrow towards the apex, and they also become shorter. It is longer toward the you can say it becomes they become shorter toward the other side, that is the afferent side. So this very side is called efferent side, and this very side is called afferent side. The lamellae are broader near the Ctenidial axis, and they become narrow toward the apex. And secondly, they are larger, they are longer in the middle, and they become narrower toward the afferent side. So this is also another thing. And secondly, you come to know that this very respiration takes place in a countercurrent way, and the exchange of the gases takes place in the through the countercurrent mechanism, wherein the blood and the water, they run in the opposite direction. The blood or the hemolymph, it flows opposite to the direction of the flow of the water. Now, when we talk about the histology of the gill lamellae is formed of the two layers of the epithelial cells, which remain separated by this very narrow space. The epithelium rests upon the basement membrane. So herein this is the basement membrane on which this very epithelium rests, and below which there is a connective tissue, and there are certain obliquely running muscle fibers and having the few scattered nuclei. So mantle epithelium, or we call it as the pallial epithelium, that lies below the muscle layer, and the gill epithelium, it consists of the three kinds of the cells. Now some we call them as the non-ciliated, epithelial cells. Others are the ciliated epithelial cells, and the few we call them as the glandular cells, because they contain certain glands as well. Now we shall be talking on the types of the Ctenidia based on the topography, that where they are present. So on the basis of their location, the Ctenidia, they are divided. They are categorized in different, you can say, terms. One, we call them as the holobranchiate, and other we call it as the merobranchiate. When the whole area is bearing the branchia or Ctenidia, we call them as the holobranchiate. If only a portion bears the Ctenidia, we call it as a merobranchiate. But this very merobranchiate is further divided into four types. That is plicate type, monopectinate type, bipectinate type, and feathered type. And this very bipectinate type is further divided as the equal and unequal, that we shall be dealing now. Now, first of all, we shall be talking about the holobranchiate type. This type of arrangement of the Ctenidia, it is found in polyplacophora. You have to keep in mind that this very kind of the arrangement is found in the polyplacophorans. In this form, the Ctenidia extend all over the body. Here in what you are seeing, that the Ctenidia that is extended all over the body. So you are seeing here that these are all Ctenidia. These are all Ctenidia. These very comb-like structures, they are usually the gills. These are the gills which are present on the all over the body. So that is why we call them as the holobranchiate. The number of the Ctenidia, it varies from the 14 to 70 pairs. So the number is remarkable here, 14 to 70 pairs, and in some cases about 80 pairs in the case of the Echinopleura. So 14 to 70 pairs and about 80 pairs in the case of Echinopleura. So in patella, that is the gastropodon, a circlet of the gill lamellae that extends completely around the margin of the mantle. So here in what we are seeing in the case of patella, that this very circlet of the gill lamellae that extend around the margin of the mantle as well. Now we shall be talking about the Merobranchiate type. The first one was the Holobranchiate, that is the Ctenidia covered whole body, that is they were present along the whole of the body. But in this very case they are restricted to the particular area. They are restricted to particular area and they can further be divided. They can further be divided. So depending upon the arrangement of these very leaflets, the gills have been divided into 4 types. Now number one, that is the plicate type. That is the plicate type. What we see here, that they are simple. They are simple. They are flat. They are transversely folded. They are projecting integumentary lamellae. They constitute the gills. And in case of the Chetoderma, what you are seeing here in this very case, in the case of Chetoderma, two large plume-like Ctenidia here in one and this very second. These very two large plume-like Ctenidia, they arise from the cloacal wall. This is the cloaca, and they arise from the cloacal wall. And the second, that is the monopectinate type. The second type, that is the Merobranchiate type. The second one is monopectinate. Now these are the flattened gill filaments. Herein what you are seeing, that these are the flattened gill filaments that are arranged in a single row. When the gills are arranged in a single row, we call them as the monopectinate type. And the examples, they are pila, they are triton, they are the examples of the monopectinate gills. Then the third one, that is the bipectinate. This one we were talking about, that is the bipectinate. Now what happens here, they can be these are the flattened gill filaments that are arranged in 2 rows. In the monopectinate there is only 1 row, but in the bipectinate there are 2 rows. They can be unequal or they can be equal. So when we talk about the unequal, the both rows of the filaments are present on the right one, are present, and the right one being the smaller in size. Now what we are seeing here in this very case as well, in the case of Haliotis, so these are the two, you can say, these are the bipectinate gills, and herein one is larger in size and other is smaller in size. That is, they are unequal. That is why they are the type of the monopectinate, where one is large and other is small, and that is why they are the unequal type of the bipectinate gills. So the another, we do have the unequal bipectinate. Unequal bipectinate, they are further the divisions or the subcategories of the Merobranchiate type. So in this very part, what we are seeing, when both of them are of the same sizes, and this is the characteristic feature of the bivalves. So when these very both rows are of the equal sizes, we say they are the equal bipectinate type. And these amongst the bivalves, they become variously modified. Now for example, nucula, we do have the nucula. What we are seeing here, they possess the short flat leaflets. Now herein I will just show you that herein what you are seeing, that they bear short leaflets, and in some forms the long filament leaflets are also present. So these filaments may be free, as in the case of arca or amusium, or may be joined by the ciliary connectives, as in the case of Mytilus as well. So in the case of Unio, the ciliary junctions are replaced by the membrane. So in poromia, the ctenidium becomes degenerated and is represented as a transverse portion. So we do have the examples here. We do have the examples here, that this is the example of the nucula, wherein I was talking about that they are the short leaflets, and in some forms they can be long filamentous as well. Now the secondly, we do have the arca. So this is the arca or the amusium, or it may be joined by the ciliary connectives. If they are joined by the ciliary connectives, as in the case of the mitylus as well, so they may be free, as in the case of this very arca. Here it is this very diagram is of the arca, wherein these are the free, these are the free, but in the case of mitylus, they are joined. Now for example, in the case of mitylus, they are joined. In the case of mitylus, you are seeing here that these are joined in the case of the mitylus. So in the case of unio, what we are seeing here in this very case, that unio, the ciliary junctions are replaced by the membrane. Herein, there is the interlamellar junctions, and the ciliary junctions, they are replaced by the membrane. In the poromyra, the ctenidium becomes degenerated and is represented as a transverse portion as well. This is the poromyra, wherein the ctenidium becomes degenerated and is represented as a transverse portion. So herein ctenidia has degenerated and is represented as a transverse portion. So we do have the examples of the nucula, wherein the short leaflets or the long filamentous leaflets are present. So in the case of arca, they may be free, and in the case of mitylus, they may be joined by the connectives. And we do have the examples, wherein we do have the example of the unio as well. We do have the example of the unio, wherein the ciliary junctions are replaced by the membrane. And fourthly, we do have example of the poromyra, wherein there is degenerated, ctenidium has degenerated and is represented by the transverse portion. So we do have all of the examples, wherein we see here all of the examples. So then we do have the last, you can say one, that is the feathered Merobranchiate type. The fourth one, that is feathered. So it assumes the shape of a feather. For example, in the case of cephalopods, what we are seeing here, that these very ctenidia, they look like that of the feather. Just like the feather of the birds, these very ctenidia look like. So these are called as the feathered type. Now we shall be talking on the modifications of the ctenidia. The ctenidia is variously modified. So I was talking that while the aquatic respiration, that is on the basis of the mode of the life in the habitat. So I told you that ctenidia are the main or the primary respiratory organs, but there are certain other, you can say, which we call them as a secondary respiratory organs in the aquatic mode. For example, we do have the anal gills. For example, here in what you are seeing in the anal gills, these are the delicate leaflet which form the rosette around the anus. So we do have the example in the form of the doris. This is the doris. So we do have this very form, wherein we are seeing that they form the rosette-like anal gills. In the Chetoderma as well, a pair of symmetrical lateral gills, they are present on the each side of the cloaca, which we have seen in our earlier slide, that the two lobes coming out of the anal portion. The second, we do have the serrata. This one, we do have the serrata. We do have in the form of the serrata, or we call it them as the dorsal appendages. So in many opistobranchs, the dorsum bears highly vascular appendages called as the serrata, and they may be simple and club shaped, such as in the case of Aeolus. They may be simple or club shaped, and in some cases they may be dendritic. So in the case of Dendronotus, so in some other cases they may be multi-lobed as well, and they resemble the bunch of the grapes as well in the case of the Doto Kika. So we do have the different modes of the, you can say, serrata or the dorsal appendages, such as highly vascular as in the case of this very serrata, or maybe simple and club shaped as in the case of this very Aeolus, or they may be dendritic as in the case of Dendronotus, or they may be multi-shaped as in the case of Doto-Kika. So then thirdly, we do have the plural gills as well. We do have the, so this is the example of the Doris. So then this is the example of the Aeolus, and then we do have the example in the form of the, you can say, we do have the plural gills. Then thirdly, we do have the plural gills. So in the case of Pleurophylida, there are certain lateral rows of the branchial leaflets that are situated beneath the mantle. So in some cases, in the case of Pleurophylida, lateral rows of the branchial leaflets, they can be situated beneath the mantle. And fourthly, we do have the pallial gills, So in some pulmonates, the secondary external gills, they develop by the enlargement of the pallial lobe just outside the pneumostome. So we do have the example in the form of the patella and Encyclidae. Herein, this is the example of the patella wherein the gills, these very gills are placed. This is also the case of the holobranchiate, but herein they are also known as the pallial gills. So we do have the patella, we do have the pneumoderma, we do have the plenorbidae. So we do have the encyclidae, and the fifth one, the fifth secondary, you can say secondary respiratory organs. Under the secondary respiratory organ, we do have the integument as well. The integument also acts as the gas exchange that occurs in certain parasitic, you can say, forms such as Entoconcha, Conia, Limpontia species, and so these are the other forms besides the Ctenidia which help in the aquatic respiration, such as anal gills, serrata, pleural gills, pallial gills, and the integument as well. Now we shall be little bit talking about the relationship between the heart and the gills. Now, what is the relationship between the heart and the gills in case of these very mollusks? So the heart and the gills are intimately related, because the main function of the gills is to aerate the blood. So the first main function is to aerate the blood, and on its way to the heart. So it is from the aeration from the gills they reach the heart. So there is a relationship between the heart and the gills. So it is the blood coming to the gills, and then it is aerated and taken to the heart. So the number of the gills, they are directly proportional to the number of the auricles. So the gills are also, they are in proportion to the number of the auricles that we shall be seeing here now. Now, for example, when we see here that when the gills are two in number, so when the gills are two in number, two auricles are present as encountered in the case of loligo. So we do see here that if the gills are two in number, then we do have the auricles also present which are two in number, as we find them in the case of octopus and loligo. So herein what we are seeing here, so octopus and loligo, we do have in the case of this very two auricles are seen. So in the case of chiton, two auricles correspond to the two sets of the multiple gills. So what we see here in the case of chiton, so this is the chiton here, what we are seeing here, that two auricles herein This is one auricle and this is another auricle. So two auricles, they correspond to the two set of the multiple gills. So this is the one set and this is the another set. So hence the auricles are also two. In the case of Nautilus, there are four gills and the four auricles. What we are seeing here, that the four gills are present. In this very case, four gills are present and the four auricles are also present. So when the gill is unpaired, the heart has one auricle, as seen in the case of Ophistobranchia, as in the case of the Mesogastropoda, and in the case of the Neogastropoda. So herein what we are seeing, that we do see them in the Ophistobranchia. In case of Ophistobranchia, megagastropods and neogastropods, what do we see? When the gill is unpaired, that is not having any pair, the auricle is seen, the heart has only one auricle. So only one auricle is seen when they are unpaired. Here in one auricle, herein also one auricle, and herein also one auricle is seen, because the only unpaired gill is present. That is why auricle is also one. Now we shall be talking. This is going to be a longer lecture, but it is obvious that we should be completing this very topic under this very heading, that is we are touching only Ctenidia. So we should be knowing all and everything about the Ctenidia. So that is why we have to complete it. So it is my request to you all, kindly bear me for some more time. So when we talk about the number, form and position of the Ctenidia, the most of the mollusks, they possess one pair of the lamellar ctenidia, and near the ctenidia are found osphadia, which tests the nature of the incoming water current. Now this is very important organ which we call as the osphradium. So the osphradium are always near the gills. So they are the important organs, and they test the nature of the incoming water. So it is by this very organ the water is tested, that if there is any abnormal changes in the water, it is tested by the osphadia. And secondly, classification of the mollusca is also based on the character of the gills. If we talk about the classification of the mollusca, that is also based on the character of the gills. The gills, position of the gills, the placement of the gills, the type of the gills, they also form an important part of the taxonomy in the case of mollusca. Cephalopods are divided into dibranchia and tetrabranchia. So that is the 2 and 4, based on the number of the gills they possess. So gastropods are subdivided into prosobranchia, ophistobranchia, pulmonata, according to the position and character of the gills. And we do have the plate-like form of the gills that lead to the name, that is the lamellae branchia in the case of the bivalves. So in polyplacophorans, the gills vary from the 6 to 80 pairs. In monoplacophora, the gills vary from 3 to 6 pairs. In the primitive gastropods, either two, that is bipectinate, for example, we do have the haliotis, the gills, or if they are the two, that is bipectinate, this is an example of the haliotis. We do have the different, you can say herein, we do have the different ones, that is in the primitive gastropods, either two, that is the bipectinate, that is found in the haliotis, or the single bipectinate gill, that is found in the patella. So in the case of mesogastropoda, in the case of the mesogastropoda, and these all, in these very cases, mesogastropoda, such as Pila, we do have the Cypria, then neogastropods such as Murex or Conus. The single monopectinate gill is present in the case of the in them. On the single monopectinate gill is present. In the case of bivalves, we do have a pair of the plate-like gills. So in the case of bivalvia, what you are seeing here, in the case of this very bivalvia, what you are seeing here, a pair of the plate-like gills are present, and in cephalopod, 4 gills in the nautilus are 2 in Colidia, that is sepia, loligo, octopus are present. So we do have the number one, that is these are the chitons. So we do have the bivalvia, then we do have the dibranchiata means 2 gills, as in the case of cephalopods. Then we do have the tetrabranchiata, these are also in the case of the cephalopods. Then we do have the prosobranchia, this is the prosobranchia. That is, what does this very prosobranchia means? Proso, it means first, that is just ahead of the heart. So then we do have the prosobranch monotocardia, and then we do have the ophistobranchia, that is the heart is first and then gills. So gills are behind the heart. Here in the case of prosobranchia, gills are ahead of the heart. So this is also led to the classification based on the position and of the gills as well. So if we talk about the number, form and position of Ctenidia. Now we shall be talking class wise. That is, first of all, we shall be talking based on the Aplacophora. When we talk about the Aplacophora, that now we shall be talking about the number, form and position of the Ctenidia. That what is the characteristic feature of the different classes wherein these very Ctenidia are present. How many number of the Ctenidia, what kind of Ctenidia and where they are located. So we shall be going through fastly in these very classes. So in Aplacophora, the mantle cavity is represented by the mid ventral. So this is the mid ventral longitudinal groove, these widening posteriorly into the small cloacal cavity. So in the case of Aplacophora, the mantle cavity is represented by the midventral longitudinal groove. So when we talk about the gills are either absent. In the case of Neomania, for example, in this very Neomania, the gills are either absent or reduced into 2 large plume-like Ctenidia. Or in the Neomania, the gills are absent, whereas in the case of the Echinoderma, we have also gone through this very, that they form the large plume-like Ctenidia on either side of the anus. So it is in the case of the Echinoderma, in the case of Echinoderma. So in the case of Aplacophora, so these are the examples of the Aplacophora, that is Neomania and Echinoderma. Either the gills are absent in the case, as in the case of Neomania, or they may be large plume-like, as in the case of the Echinoderma. If we talk about the Polyplacophora, the class Polyplacophora, the bipectinate Ctenidia, they are located in the mantle groove. Either the number of the Ctenidia, they vary from the 14 to 80, as in the case of Acanthopleura, or the gill rows may be holobranchiate, or they may be holobranchiate with 2 exceptions, bearing the merobranchial type, as in the case of this very chiton levis. They may be holobranchiate or they may be merobranchiate. So if we talk about the number, form and position of Ctenidia in the case of Monoplacophora, what we are seeing here, the pallial groove contains 5 to 6 pairs of the unipectinate gills. So what we are seeing here, 1, 2, 3, 4, 5. So 5 to 6 pairs, 5 on this very side, 5 on this very side. We are seeing here that the 5 to 6 pairs of the gills are the unipectinate gills. They are present on either side. So we call it as the neopalina. Example is neopalina. So this is the example of the Monoplacophora. So when we talk about the gastropoda, this is the important one, where in the number, form and position of the Ctenidia, they differ widely. So first of all, we shall be talking that the gills vary widely in number and position in this very group. We have the prosobranch. Now this is very important to learn. that what prosobranch means when the Ctenidia lie in front of the heart. So when the Ctenidia, so herein I will just try to show you that herein these are the gills, these are the gills here, and they are just in front of the heart. So when the gills are in front of the heart, they form the prosobranch. So in this category, we do have the diatocardia, and we do have the monotocardia. So when in this very case, in the diatocardia, the arrangement is the most primitive. The gills are too long here in this very case. In this very case, um, what we see that the arrangement is most primitive. The gills are two, long feathered on each side and lie symmetrically to the middle line. So we do have the example in the form of the fissurella, and we do have the monotocardia. We do have the form of the monotocardia, wherein arrangement of the gills that is remarkably uniform, and the single gill feathered on one side and united to the mantle along its whole length. For example, in the case of this very triton, in this very case, what we are seeing here, the arrangement of the gill, it is uniform arrangement of the gills, and this is on one on this side and other on this very side, and a single gill feathered on one side and united to the mantle along its whole length. So example is the triton. So we do have the example in the form of the triton. Then we do have the opistobranchs. In the case of opistobranch, what we usually see that the gills lie partially in the mantle cavity, or they may be positioned just behind the heart. So here in what we are seeing that these are the gills, which in some cases they may be nudibranch, that the gills open, that is not covered. So here in what we are seeing that these are the gills which are just behind the heart. That is why we call them as the opistobranch, such as in the case of aplysia. Then we do have the another class, that is the bivalvia. So we do have the different number, form and position of Ctenidia in this very class. The first of all, we shall be talking about the protobranchiata. So in these very bivalvia, the gills are bipectinate mostly, and they are equal on either side and usually very large, having assumed the food collecting function in most species in addition to the gas exchange. So when we talk about the protobranchiata, first of all we shall be talking about this very protobranchiata. Gills are the smallest. In this very form, the gills are the smallest and lie behind the foot at the back of the mantle cavity. So in this very case, nucula is the example. So in this very case, what we are seeing that these very gills, this is the enlarged view, open view of the Ctenidia, and this is inside the organism. What we are seeing here, that these are smallest and lie behind the foot at the back of the mantle cavity, and the example is nucula. Then the second one we are seeing here, the filibranch. This one, that is the filibranch. So the what we are seeing here in this very case, each gill is separately this forms a W-shaped letter. This forms the W-shaped you can say section with the long narrow limbs. So the gill axis lies at the middle angle, and the examples, they are the arca, mytilus, and anomia. So these are the example of the filibranchs. So we do have these some examples of the pseudo-lamellae branchiata, and they are reflected by the dorsal tip of the gill filament, and have which have coalesced laterally with the mantle and mesially with the base of the foot. So in the pseudo-lamellae branchiata, the reflected dorsal tip of the gill filaments have coalesced laterally with the mantle and mesially with the base of the foot. So the gill has the greater cohesion than that in the case of the filibranchs. So the examples, we do have the astridae, then we do have the pactinidae, we do have the plerichia. Then we do have the eulamellae branch. example in the form of the Lamellae branch. The adjacent gill filaments, these very adjacent gill filaments, are united by the vascular cross connections. They are united by the vascular cross connections, leaving narrow opening, that is the very narrow opening in between, that is the ostia, very narrow opening in between. So two lamellae of each branch, they are attached back to back in the same way. So we do have the example of the cardiacia and mycia. Then we do have the septibranch. We do have the septibranchia. The Ctenidia do not exist as such. Herein we are unable to see the Ctenidia. We are unable to see the Ctenidia here, and they actually they are converted into the horizontal muscular septum running from the base of the foot to the mantle and extending right back to the we can say the siphons. So the example is poromia. So we do have the example in the form of the poromia. So another class, that is the Scaphopoda. In this very class, the gills are lacking, and we do have the tusk shells in this very case, wherein the Ctenidia are absent. Then we do have the Cephalopoda, the important one, that is Cephalopoda. The gills here in this very case, what you are seeing, this is the gill, this is a plume-like gill. The gills are large, they are paired, they are bipectinate, what you are seeing here, and they are suspended on either side of the rectum by the afferent edges. And then secondly, the gill filaments, they are firm and fleshy, they are non-ciliated and thrown into primary and secondary folds to increase the surface area. So, in the case of Nautilus, the gills are two pairs. The hemolymph, it flows through the gills and is assisted by the pulsatile accessory branchial heart, which is located at the base of the gill in the course of the afferent ctenidial vessel and placed in an you can say annexi of the pericardium. Then we do have the pallial contractions which drive water between the gill filaments at the great pressure. So we do have these all these very things studied, that is how these very number of the ctenidia, they how many numbers of ctenidia are they present in different classes, how many forms of the Ctenidia are present, and what is the position of the Ctenidia in different classes. So next time we shall be dealing with the terrestrial mode of respiration in the case of the molluscans. We could not complete this very lecture in almost 1 hour, but next time we shall be dealing with the terrestrial respiration. So it is my request to you all again to like and subscribe my channel Biolearnia for new lectures. Thank you very much.